distinguished teachers, colleagues and students, I'm here to share with you a dream plan and ask this question: can we realize this? The dream plan is, how do we get rid of disease and disorder from the planet? Well, one possibility is computers should help us in this endeavor. The related question is, how is chemistry related in this computer-aided drug discovery? And another important question is, do we have anything like this happening in India, an indigenous software which can help us eradicate disease from the planet? So let's proceed to understand where are we today. Well, today we talk of big data. What's the big challenge here? Well, the big data should lead us to information, to knowledge, and eventually to products useful to society. So what kind of big data is available and how can it help us get rid of disease? Well, one area is a National Center for Biotechnology Information, which gives you huge amounts of genomic information of thousands of organisms. Another area is that of protein sequence information. Again, UniProt is a database which hosts millions of protein sequences. Then there are structural databases called Protein Data Bank, and there are small molecule databases also relevant to chemists, about 35 million purchasable compounds. With so many compounds available, how is it that diseases and disorders are still prevalent? And that's a big question. So let's see how we use this big data, make some drug molecules and get rid of diseases. Well, some milestones in this drug discovery area. All of you are aware during the World War times, antibiotics surfaced and they became very popular. Some of them were discovered in a hit and trial approach, and others serendipitously. Prontacil was one of those antibacterial compounds, and penicillin, all of you are aware of the history of evolution of penicillin. So, and these scientists of course were awarded the Nobel Prize. So in those years, 1945 Nobel for penicillin and 1939 Nobel for Prontacil. And interestingly, Dorothy Crawford Hodgkins got the Nobel for elucidating the structure of penicillin. It has interesting chemistry, beta-lactam ring and so on. She also solved several interesting macromolecular structures, including structure of vitamin B12. Then Nobel for streptomycin followed. That was in 1952 to resist towards tuberculosis. Then more recently, all of you are aware, malaria found a cure in artemisinin as from a bark, and U22 is the Nobel laureate who made it happen, and similarly for ivermectin as well as artemisinin. Both these were awarded the Nobel in 2015. Now I've been listing lots of Nobel Prizes. So, well, what's the idea of all this? Well, taking a departure from the traditional hit and trial approach, can we do a more rational approach? That is, can we focus on the biochemical process of disease, look at the physiological process and look at the biomolecular target, and then start thinking of what chemical can interrupt this process so that the person becomes normal. So that kind of idea was initiated by these scientists, Black, Alien and Hitchings, and this was in 1988 Nobel, and on focus on the process for drug discovery. Then 2009 of course has seen Venki Ramakrishnan's contributions along with his colleagues Tom Stites and Ada Yonath. They put together crystal structure, huge crystal structures consisting of hundreds of thousands of atoms of our ribosomes in bacteria, and these ribosomes are different from those in humans and animals. So now if you make these ribosomes as your drug targets, bacteria cannot replicate, whereas animals and humans are safe. In that context, you can say, well, here is an opportunity to generate antibacterials. So the structure then is a starting point for drug discovery. Now to talk of the rational drug design as to what do we do today, and essentially you need a structure, and drug molecule is like a duplicate key. If your protein is an artificial, is a lock, you want to jam the lock, that is like an inhibitor, or you want to open the lock, that's an activator. So your drug molecule is either an inhibitor or it's an activator. Now why protein? Most of the available drugs in the market today target proteins. Proteins are easier to handle. The molecules that you make to target these proteins as inhibitors are small. It's not to say that DNA or RNA cannot be targets, but someday with progresses in nanobiotechnology and drug delivery systems, one would expect that even DNA could be a popular target. But as of now, proteins are the most attractive and important targets. And most of the drugs act as inhibitors. There are fewer activators as drug molecules. Now, what is my dream plan in this? Let's go back to our first description of what is a dream plan. Suppose you had genomic information of an organism, let's say a virus. How do you make a drug molecule? What's the pathway? Can we sketch a pathway? I call this as a country path today, but tomorrow it will be like an expressway, Delhi to Agra with 16 lanes and cars zooming past and so on. How do we sketch this boulevard? So I start with the genomic information, and from the genomic information, I decode this and put together the protein coding regions. So I have M, A, L, W, M, etc. So those are my protein sequences, and then I build a three-dimensional structure of the protein, as you can see, the 3D structure. And after I have the 3D structure, I unleash this chemistry-based idea of making a molecule which has complementary interactions to the protein. So the idea is maximize these interactions between the small molecule and my protein target and ensure that it binds as opposed to the natural substrate for the protein and then stays there. So you have designed now an inhibitor molecule. Of course, it has to pass several safety tests such as toxicology. It should not be toxic to the human host, and it should be easily synthesizable, it should be available, and bioavailability. There are several other little filters that one can do. Now most of this can be done on a computer, and once you have this, you can have a panel of doctors not necessarily sitting geographically in proximity, but they could be all over the world, and the computer will email them your molecule and the target and the patient's symptoms and the records, and eventually the panel will say, well, this is the molecule, this is the dosage, and then the person is administered that, and he or she is running happily, jogging and so on. So 10 a.m. the disease is reported on a smartphone, 5 p.m. the drug is delivered at the doorstep. Well, that's my dream plan, and looks like this is feasible. Well, what is it that has to go inside? Well, obviously genomics, proteomics, metabolomics, information technology, and don't forget chemistry. Without chemistry, well, there's no drug molecule. Now, where is the need for drug discovery and what's the urgency about it? Now, I'm here referring to the WHO report released recently. You can see in this the pink color almost all those favorite antibiotics that you might have administered or taken as a little kid, small kids and so on. So what's interesting in this is penicillin, of course, is there, and so also the more recent generation rifamycin, etc. They are all out there. But beyond 1987, you see a discovery void. There is hardly any new antibiotic coming in the last 13 plus 20 now 35 years. And so that's very strange. There is a discovery void. What does that mean? It simply thinks that we are very complacent. We think we have conquered bacteria. Now, unfortunately, bacteria are becoming resistant to the existing drugs. So that's a danger. That's a serious situation, and no new antibiotics are coming up on one side. On the other side, bacteria are becoming resistant to these drugs. So what can this AMR, antimicrobial resistance, do? Well, it doesn't have to be a deadly disease to kill people. It could be simple fall from a bicycle of a little kid, and that could be that little scratch on the hand can be a colony for bacteria. And since none of the antibiotics is working, the kid can die. That's a serious disaster situation if we don't discover. So let's commit ourselves to discovering some new antibiotics, develop new antibiotics, and keep some of them in store so that bacteria don't know, right, that you have already some newer weapons in your arsenal against the bacteria. Well, why is it that people have not been doing it? Well, they have been doing a lot of drug discovery over the last 80 years, but pharmaceutical R&D is very expensive. Now, if you focused on the number of drugs developed over the last 80 years, and here I am showing the statistics from Food and Drug Administration. NME stands for a new molecular entities. How many molecules have come about? Well, I already referred to the zinc database, which has about 35 million compounds. Some other databases are reporting 150 million compounds, and you can see most of the organic laboratories all over the world are busy making molecules and molecules. But how many molecules have become drugs? In the last 80 years, only 2,500, just 10 to the 3 versus 10 to the 7 or 10 to the 8 that are available. Compound space that's available is huge, but number of drugs is very, very small. This is to say that we don't have enough diseases. No, we have enough diseases and disorders to worry about. Molecules are hard to come by. Now, why would that be so? Well, that's because drug discovery is expensive. On an average, it takes about 14 years and about $2.6 billion. So that's a lot of money. So most of the companies cannot afford this kind of money. This is not to say that every drug in the market takes 14 years and 2.6. These are average numbers. So it's expensive and it's time consuming, and there are not too many investors. And this is particularly true for third world country diseases where the investments are low, but the need is very high. So now what do we do? Well, here is what our famous singer says, "Aapka kya hoga Janabe Ali." Now, what does this mean? Well, it means that you find new targets, develop new drugs, and keep some in store, and don't tell microbes you got them. So that's the general idea here. Now, what is it that we are doing now at IIT Delhi in my laboratory? Well, we have genomic information, and from genome, you give me a genome, I will tell you which parts of the genome code for a protein, and that software is called ChemGenome, as you can see. Now, once I have identified parts of DNA which code for a protein, I then write down the protein sequence, and that's very easy. Hargobind Khurana told us that 3 bases code for 1 amino acid. So, using that simple genetic code, I can write down the sequence through mRNA, DNA to mRNA to a protein sequence. After I have the protein sequence, comes the grand challenge problem of protein structure, because I need the 3D structure. Remember, to develop a drug molecule, I need the structure of the shape of the lock or the structure of the protein. So, I have to now develop a 3-dimensional structure starting from the protein sequence, and that is where my software Bhagirathi is going to help. And beyond that, I have the structure, now the target. So, the target could be DNA, or it could be mRNA or microRNA or any one of the other RNAs of interest, or it is a protein. Now, I want to come up with a molecule. That molecule is target oriented or it's target specific. So, that software is called Sanjeevani. So, I will share with you on how do we proceed in this context on this genome all the way to a hit molecule or a drug molecule. I will call modestly it as a hit molecule. So, first phase is a hit, next phase is a lead, and the third phase of course is a drug molecule. So, how do you go from a genome to a hit molecule? Well, one of my colleagues gave a problem to me. He said, can you come up with a new molecule against hepatitis B virus? And this is of course a serious need, and the virus has developed resistance, and the carriers are increasing. So, I asked him, anything known about this particular virus? He says, well, something about it is already known. It has about 4 ORFs, open reading frames. It can code for some of these proteins, and the function of the proteins is also known. But I want a molecule which is not a nucleoside analog, because patients have reported resistance against nucleoside analogs. So, how do I now target a protein that's not a polymerase and that's not a nucleoside analog? Now, can you do something about it? So, this is a question that he posed. I said, well, it's very easy for me. Just give me 14 years and $2.6 billion, and I will definitely give you a molecule. Well, I'm serious. More seriously, I said, just give me 3 hours. I can make this happen within India on my computer at IIT. How is that so? Well, let's go over back to that big data slide where NCBI has all the information on the genomes. Now, I go to NCBI site, I download this hepatitis B virus genome. So, complete genome is available on my laptop. Then I push this to my ChemGenome software, and this chemistry based genome annotation software. What does it do? It identifies 5 protein coding regions. Once I have these 5, I could have taken all the 5 as my drug targets, because these are viral proteins. They do not share similarity with human proteins, luckily. So, any one of them could have been a good drug target. So, my friend suggested focus on protein 2. So, I did that, and then this is my sequence. So, sequence is that MQL that you see here, methane, and glutamine, leucine, phenylalanine, etc. So, that is my protein sequence. Then I take this and then pass it on to my Bhagirath Edge software. So that gives me a three-dimensional structure. Then after that, what do? Then I start scanning this protein for binding pockets, potential functional sites on the proteins. Remember, I don't have any experimental knowledge. I started with the genomic information. From genome, identify the protein coding regions and then sequence, and from there to structure. And now I'm looking for the so-called active sites. So once I have this I can do this with my Sanjeevani software. 10 potential binding sites are identified. Then I take million compound library. Remember, we already have 35 million purchasable compounds. So why not utilize that knowledge of compounds which are already out there? So if any one of them clicks, it works, then you can easily purchase them and then you can validate them in a biology laboratory. So that's a general idea here. You could, of course, design a molecule. This is not to say that we cannot appeal to newer, creative, synthetic chemistry. It's possible, very much possible. So let's scan a million compound library against each of these 10 potential binding pockets. Then what do we have? Well, we have a smaller list now, about let's say 2,000 molecules which are available, and with the good binding energy. Then I proceed to for atomic level docking and scoring. Now, when I say atomic level, I'm looking at these electrostatic interactions, van der Waals interactions, and hydrophobicity, ensuring that my delta G, my binding free energy, is the highest for the molecule that I'm predicting as potential molecules or hit molecules. So after I do the atomic level docking and scoring, that's essentially ensuring that the drug molecule that I have selected, or the hit molecule that I have selected, is interacting very strongly with the protein target. I shortlist a few of them for the purposes of procurement or synthesis, and then ask my biology friend to test it. And luckily for me and for my student, they did the testing, they procured the molecule, they did the testing, and they showed very good binding with the protein. What you are seeing are molecules 6 and 7 showing micromolar activity, micromolar compound. And remember, these are dissociation constant. This is a convention for reporting IC50 values. So if the dissociation constant is low, the binding is very high. So for example, a nanomolar compound is a good, extremely good compound. A micromolar compound is a very good start, right? So that's the idea. So now with current days computer aided drug discovery methodologies, what I can say is a micromolar compound is a routine matter. I can start from a genome or I can start from a protein. I need a target. Once I have this target, then making a molecule using basic principles of chemistry is not that difficult. What is difficult is how do I optimize this further to a nanomolar compound? Well, my medicinal chemistry friend should help, and so also my physical chemistry friend should help in this process. But what is even more interesting is how do I make this non-toxic to humans? So that's a very challenging problem. We still don't have a good molecular picture of what is toxicity due to. If you can figure out, or if you can come up with 5 empirical rules of toxicity, then life is made simple. I don't need $2.6 billion and 14 years of research to come up with a drug molecule. So what I presented to you is a genome to drug pathway. As I said, this is a country path, and hopefully tomorrow this will be automated. The entire thing will be automated, provided you can fix that toxicity issue in this picture, right? So that's the methodology that I presented to you, the genome to hit molecule methodology. Now I'll focus on the details of where is chemistry playing a role in this drug discovery, in this computer-aided drug discovery, or more specifically structure-based drug discovery. This Sanjeevani is an indigenous endeavor. It's an ongoing methodology started way back in 2000, and now it has matured to a stage where it has several success stories. I'll share with you a few of those success stories. So what's the overall plan? How do we proceed? Well, either you have a database like the million molecule compounds, or you are going to draw the molecule on the screen yourself. You will create a molecule on the screen yourself because you already know these are the interactions that I want. I want a couple of hydrogen bonds here. I want a non-polar interaction on this side, and so on. So you could generate a molecule yourself. So you have a candidate molecule. Then preferably you pass them through what are called drug-like filters, such as bioavailability And here there are some rules already available, such as Lipinski rules, which enable you to discriminate non-drug like molecules from drug like molecules. So that will be a good safe filter to start with before you go on to more compute intensive processing. Then after that, what do we do? Since you have drawn the molecule or you have adapted the molecule from a database, you may want to do a geometry optimization. Now there are force fields available, molecular mechanics force field, or you can also do quantum mechanical calculation and quantum mechanical optimization of the geometry, derivation of charges. All these will become important when you have to assign force field parameters such as delta plus, delta minus, those partial charges on the molecule that you have to assign to calculate the interaction subsequently. And then the next step will be, of course, put in the three-dimensional structure of the drug target. If you have it, fine. If you don't have it, you can always generate, and we have enough software such as Bhagirath available to develop three-dimensional structures of the protein target. We can also do a DNA target very easily. So having obtained the 3D structure and your drug molecule with parameterized, now you are ready for active site identification and docking. Docking is one of the most challenging problems in drug discovery, computer-aided drug discovery, right? Because you have a huge set space. So what one does is typically take recourse to rigid body docking, where you're searching in the 3 translations and 3 rotations about 6-dimensional space. You can put in flexibility of the ligand as well as that of the target, that is amino acids or nucleic acid bases, subsequently through a molecular dynamics simulation. So after doing the docking, then you want to score. Having docked, what is the best score, which molecule scores the highest, etc., in terms of binding energy. So we can do a binding free energy estimate. So that's called scoring. So after you've done the docking and scoring and you rank the molecules, take the best molecules and then do a molecular dynamics simulation, essentially solving F equals to ma on the protein, the ligand, and the protein ligand complex. And then you can do a post facto free energy analysis and understand which molecule is most likely to be promising, which will stay. So during your MD simulations, you can mimic as much as you want of the real cellular milieu as much as possible. So put that in so that in real life, when you predicted the molecule, it works in real life as well, in the test tube as well. So that's the overall strategy. So this is what we proposed 2006, and now we have elaborated it much further. Now the latest version of Sanjeevani can handle millions of compounds. We also have a non-redundant database of small molecules, or you can do self-draw the molecules. So again, I repeat, we check the Lipinski compliance, that is for bioavailability, drug-like or bioavailability filter. Then we also have put in this RSPD methodology, a quick scan of 1,000,000 compounds. Remember Dhanvantari pathway that I was mentioning to you? I did this screening not in 14 years, but in 3 hours. How can you scan millions of compounds? Well, it's possible these days using supercomputers and QSAR technologies. So we have developed a QSAR equation which takes care of number of hydrogen bond acceptors, donors, and nonpolar groups, and so on. And without atomic level docking, I would scan these 1,000,000 compounds to see what are the most promising hit molecules. Once I narrowed them down, then I go for the more compute intensive atomic level docking, scoring, etc. And beyond that, to even more compute intensive molecular dynamics simulations, and ultimately I have ranked them based on free energy. So that's the overall scheme. So here is a software which is available, Lipinski rule of 5. So you can upload your molecule and it will print out the properties of your molecule. Then active site identifier, active meaning that you upload your target, biological target, and this software will tell you these are the binding pockets, this is where it's going to bind, these are the atomic, the residues that are involved in these interactions, and so on. Of course, we did validation of the software against several known softwares already available, and what we find is that if you focused on top 10, remember I was mentioning to you that I took about 10 binding pockets and I'm scanning molecules against all the 10. The confidence level is very high when you have 10 binding pockets. But if you already have some biochemistry information, some experimental knowledge on where is the binding pocket, what are the active site residues, then you can zoom in on that out of these 10 automatically. So this particular one is working quite well. And then we have a 1,000,000 molecule scanning. So this is very popular with the users these days. Now what it does is that you upload your target and it will do a 1,000,000 molecule scan within minutes, and it will give you identify, well, these are the candidate molecule that you should process further. So that is the idea. Now, while I've been focusing a lot on target and a molecule, traditional medicine like Ayurveda, however, does not do one target, one molecule. It gives you a concoction. So the challenge is, what are the various targets which are affected by this pool of small molecules in the concoction? Well, that's a good challenge. So if you are trying to bring in allopathy and Ayurveda on the same platter or same platform, what you would like to know is, which molecules are there in my extract like tulsi extract or neem extract, and what are the biological targets, which proteins are they binding to, and how are they metabolically related to prevent or cure a disease. So this software, BioTalks software, helps figuring out the bioactivity information of organic compounds. So we've been now trying to use this extensively in trying to understand at a molecular level as to how does this Ayurvedic medicine work. So this would contribute. Western medicine is all about one target, one drug. Can you do multi-target, multi-drug in the traditional way? Traditional medicine has been so successful, so how do we bring them together on parts? So that's the idea here. So now back to targets again and molecules. So once you have a molecule, you obviously have to do Schrodinger equation calculations. So you have to do geometry optimization, assignment of partial charges for these interactions, Coulombic interactions between the molecule and the protein target or the DNA target, right? And then automated docking scoring methodologies. I already mentioned to you, this is a docking molecule, and then here is an automated docking. So based on the binding free energy of the molecule, you can figure out where is the binding pocket. So that's the idea of this particular software, and we are comparing now the accuracies. So you're within 2 angstrom. Suppose you're printing a complex structure on the computer, and now you want to compare, is this really good? Well, you have crystallography, protein ligand complex, and you can compare and calculate RMSD, root mean square deviation of each of the atoms, and what it shows is that these computer aided docking methodologies are working just fine. They're really reliable. And in terms of scoring, well, I'm showing you pictures of about 100 protein drug complexes as well as DNA drug complexes, the predicted versus the experimental binding free energies, and the correlations are reasonably good, right? And of course, comparison with some of the existing scoring functions. And if there are metal ions in the protein, no problem. We can still do a quantum calculation and then evaluate the charges, and then use the traditional docking scoring methodologies with the metal ion as a part of the protein target. So here are some softwares which are available like BAPL, BAPL Plus, and this Paradox methodology. Now, of course, machine learning is becoming extremely popular these days, and my students don't want to stay away from this new ideas, new trends coming up. So what they have done is to take our BAPL, that is binding energy methodology software. So they parameterize the van der Waals, the electrostatics, etc., using machine learning, random forest to be more specific, and then they optimized it and they evolved a scoring function which is supposedly the best scoring function now globally. So that's just coming up, and very soon we'll make that happen. I will make that available to the user community very shortly. So then a question that comes up is the drug molecule going to stay a drug molecule inside the body? Because there are cytochromes inside the body, and these can chew up, they can oxidize the molecule, they can fragment them. So one can, of course, prevent this from happening by doing a sites of metabolism prediction, and that requires some quantum calculations again of the HOMO, LUMO, and correlating them with what are possible sites that can be cleaved and what products can occur. What you want is prevent the cleavage of these products. If you are looking at a prodrug, it's a different story. But if you want to look at a drug molecule, you don't want it to be cleaved. So you want it to remain so that it goes and binds to the target and elicits its drug response. So that's possible, and this software is now available, right? And then I mentioned to you some time ago that proteins are attractive targets. Now with the genomic information, so if you want DNA to be a drug target, which is probably a reasonable thing to expect, especially in cancers, and you want to know you want the molecule which will bind only to DNA of this particular organism, let's say malarial genome or Plasmodium falciparum genome, or a particular stretch which is causing this cancer. Now, what should be the size of this DNA, which is your drug target? Well, doing simple comparative genomics of human genome versus a Plasmodium falciparum genome, we find 18 to 20 base pairs is unique, meaning that you won't have side effects if you were to if you were to block for 20 base pairs on the DNA. Now, 20 base pair coverage will be axially about 68, 34 times 2, 68 angstroms, and that would mean radially it's a very long, big polymeric molecule. So, obviously, drug delivery will be a problem. And remember, DNA is inside another membrane, nuclear membrane. So, you have to cross an extra membrane, you have a huge molecule to target. But again, nanobiotechnology and drug delivery systems and it should help us to overcome these kind of problems in the near future. But this is not to say that we don't have methodology. We have a predictive methodology which is very popular in the area of DNA based drug discovery, minor group. We also have something for the intercalators, that is, these molecules bind between the bases as they intercalate into the DNA. So, we could have either intercalators as the drug molecules for the DNA or minor group binders as drug molecules for the DNA. Now, quickly on the free energy side, once you have a molecular dynamics trajectory, that is F equals to ma on the protein, the ligand and the complex, you can set up a simple thermocycle and figure out what's the free energy of binding. Delta G naught equals to minus RT logarithm of K. We are focusing on that K, that equilibrium constant, right? And if to get that equilibrium constant and then eventually to compare that with the experiment, you would need this delta G. Then one can do a trajectory analysis of these MD trajectories and evolve these free energy components, and then do a systematic analysis and preferably optimize the drug molecule so that it shows better binding affinities. So, what more can we do with computer-aided drug discovery beyond optimizing a molecule which will bind very tightly to the biological target or biomolecular target? Well, we can also focus on elimination of this molecule to non-targets so that we don't have toxicity arising because of off-target binding of the molecule. How is it possible? Well, if you have the structures of all the proteins inside the human body, for instance, let's say 20,000 proteins, then you take this drug molecule 1 and then you do the docking against all the targets. Remember, you want, for example, drug 1 to go to target 1. So, what I am showing is a blue square there, meaning it is binding with high affinity. Now, what does the drug 2 do? Well, drug 2 is supposed to bind to target 2. Similarly, drug 14 binds to the last one binds to target 14. Now, if you see these blue colors at wrong places, for example, drug 14 is showing a blue color not only for target 14, it is showing for target 11, target 9, meaning that it's doing off-target binding. So, this is a bad drug. It's not going to be a good molecule because it's going to bind to every protein promiscuously. So, you can screen out such molecules and ensure that your drug works like a magic bullet and goes to the target that you are interested in. And computers can help you do this job of eliminating such molecules. So, now briefly some success stories of the Sanjeevani, this so-called or indigenous software, and here we have designed some molecules, biphenyl compounds, and estrogen receptor is a drug target, and we did get some molecules on the computer and the experiments validate them, and they say clearly that you have come up with an estrogen alpha molecule with micromolar affinities or dissociation constants. And for Alzheimer's, acetylcholinesterase was the target, and here we have again some molecules which are binding very efficiently, and experiments are validating that these are all low micromolar compounds, which is very good news for drug discovery. Then antimalarials, again we have trizine based molecules and they are binding to this N-methyltransferase, which is a unique target for the falciparum, that's malarial species, and it's non-existent in humans. So, these molecules then should be safe as drugs, potential drugs. Right now we are saying they are good candidate drugs. Some processing is still further required to make them into a drug molecule. And here I am showing you antivirals. So, Sanjeevani designed these molecules, molecule 6 and 7, and they are showing very good binding affinity to the HIV virus, and not only to HIV, to HRV as well. So wherever there is a 3C protease, a P coronavirus 3C, and probably these molecules are going to work. So essentially it's like a generic drug molecule for a battery of these viruses which share common features at the protein structural level, right? And people say nature is a good idea, so Sanjeevani has entered nature in 2016, and here this is a collaboration with Harvard Medical School, and this is for the development of an antifungal molecule. And we've also been doing a lot of drug repurposing in collaboration with our colleagues in the National Cancer Institute, and hoping to see some more molecules coming out of Sanjeevani, right? And so I summarized you as to how do you go from a genomic information or protein using Bhagirath, or structure using active site finder, or if you have all this information already and molecule is known, drug is known, target is known, you want a better molecule, well, you can start off with RSPD again, do a scan or design a molecule which has the optimal interactions, and do a docking scoring, and eventually come up with a better molecule than you anticipated. Now, well, in today's world, if these molecules are working and these softwares are working on a big supercomputer, I don't think anybody is impressed. You would want it to be on your mobile phone while you are traveling in the metro or somewhere else. Well, luckily for you, our students have developed a mobile app, Sanjeevani app, on your mobile phone. You can easily download it and then do drug discovery, computer-aided drug discovery while you are walking, talking, and so on. So that's feasible. So what is the future of drug discovery? Well, I would say that ADMET, that is absorption, distribution, metabolism, excretion, toxicity. ADME is under control. T, the last word, the last letter, is not in control. So what are those 5 rules of toxicity? We have some ideas on what toxicity can be due to, but it's not fully conquered yet. As affinity specificity from a delta G point of view is taken care. What is not taken care of as yet, and why this drug discovery endeavor is taking such a long time, like 14 years and $2.6 billion, is because of toxicity. So if you can take care of toxicity, I think you have helped mankind in a big way. So that's the team that's made this possible. 50 teraflops, 200 terabytes of storage, and some smart dedicated student power, and several people. And in fact, I summarized to you the work of 25 PhD students. What's interesting is I keep telling my students that they should do this chemistry and drug discovery with love, with passion for science and for humanity. And what I find is that there are several love marriages in my laboratory. So that's the good news part. Well, thank you so much. Again, I'll be very happy if you send an email to me querying with your queries or with your answers or observations, and I'll be happy to answer. Thank you.